Every single minute, the equivalent of one garbage truck full of plastic is dumped into the ocean. Every minute. By the time you finish this sentence, another truckload is gone. Into the water, into the food chain, into the fish, into you. There are currently 150 million metric tons of plastic in the ocean. By 2050, there will be more plastic in the sea than fish. By weight, more plastic than fish. That's not an activist's estimate. That's the Ellen MacArthur Foundation and the World Economic Forum. Those are not radical organizations. And in a lab at Yale University in 2011, a group of students on a research trip to the Amazon rainforest discovered a mushroom that eats plastic. Eats it, breaks the polymer chains apart, digests polyurethane, one of the most common and most persistent plastics on Earth, and converts it into organic matter, into carbon, into soil. The mushroom doesn't need light, it doesn't need oxygen. It can function in a landfill, in the dark, underwater, in the exact conditions where plastic currently sits and waits to decompose for 500 years. This discovery was published, peer-reviewed, confirmed by multiple research groups, covered briefly in scientific media, and then nothing. No emergency funding, no global cleanup program, no front-page headlines, no industrial deployment. 13 years of nothing. I need to show you why. In 2011, a group of Yale University students traveled to the Ecuadorian Amazon as part of the university's Rainforest Expedition and Laboratory program. The trip was led by Professor Scott Strobel, a molecular biophysicist. The goal was to collect endophytic fungi, organisms that live inside plant tissues, and test them for useful biological properties. One student, Priya Anand, isolated a fungus from a plant specimen that did something no one expected. The fungus could break down polyurethane, not slowly, not partially, completely. The species was identified as Pestilotosius microspora, and its ability wasn't a minor curiosity, it was unprecedented. Let me explain what polyurethane is and why this matters. Polyurethane is everywhere. It's in your shoes, your furniture cushions, your car seats, your insulation, your refrigerator, your mattress, your phone case. It's one of the most widely produced plastics in the world, over 27 million metric tons manufactured every year, and it is extraordinarily difficult to break down. In a landfill, polyurethane persists for an estimated 400 to 1,000 years. There is no natural process, no weathering, no bacterial action, no chemical exposure under normal conditions that efficiently degrades it, until Pestilotosius microspora. The Yale team published their findings in the journal Applied and Environmental Microbiology in September 2011. The paper documented that this fungus could use polyurethane as its sole carbon source. That means the fungus doesn't just tolerate plastic, it feeds on it. Plastic is food. And here's the detail that should have made this front-page news worldwide. The fungus could do this anaerobically, without oxygen, in the dark, in conditions identical to the bottom of a landfill. Every other biodegradation process science had discovered up to that point required aerobic conditions, light, surface exposure. Those conditions don't exist in the places where plastic accumulates. Landfills are anaerobic. Ocean sediment is anaerobic. Underground dumps are anaerobic. The places where plastic lives and never dies are exactly the conditions where this mushroom thrives. The lock and the key, found on a student research trip in the Amazon. The Yale discovery was extraordinary, but it was only the beginning, because once scientists started looking, once someone proved that fungi could eat plastic, researchers around the world started finding plastic-eating organisms everywhere. In 2017, a team at the Kunming Institute of Botany in China discovered Aspergillus tubangensis, a common soil fungus found on garbage dumps in Pakistan that could break down polyester polyurethane in weeks, not centuries, weeks.
The fungus secreted enzymes that degraded the polymer bonds and used the carbon for growth. In 2018, researchers in the UK identified a mutant enzyme derived from a bacterium found at a Japanese recycling plant that could break down PET plastic. PET, polyethylene phthalate, is the plastic in water bottles, food containers, and polyester clothing. It represents approximately 70 million tons of global production yearly. The enzyme broke it down into its original chemical components, which could then be used to manufacture new plastic, a complete recycling loop, biological. In 2020, a team at the University of Sydney documented over 400 species of fungi and bacteria with demonstrated plastic degradation capabilities. 400 species. Found in oceans, in soil, in landfills, in compost heaps. Nature had been evolving solutions to plastic for decades. We just hadn't been paying attention. In 2022, researchers at the University of Texas at Austin used machine learning to engineer a supercharged version of a plastic-eating enzyme that could break down PET containers in as little as 24 hours, one day. A plastic bottle that would sit in a landfill for 500 years, digested in a single day. And then, most remarkably, in 2023, scientists confirmed that certain fungi don't just break it down, they convert it into biomass, into organic carbon, into the building blocks of soil. The plastic goes in, fertile organic matter comes out, waste becomes food, pollution becomes soil. The science isn't emerging, it's arrived. It's sitting on the table, published, proven, confirmed by universities across 4 continents over 13 years. The discovery phase is over. The question now is why deployment hasn't started. Now, the question that every single person watching this video should be asking: if we've known since 2011 that fungi can eat plastic, and if we've known since 2023 that fungi can convert plastic into soil, why is there still a garbage truck of plastic entering the ocean every minute? To answer that, you need to follow the money. And by now, if you've been watching this channel, you know that this is where the story always goes. The global plastic industry generates approximately $600 billion in revenue per year. That's production alone. Add plastic waste management, collection, transportation, sorting, landfilling, and incineration, and you're looking at an additional $50 billion per year. The recycling industry adds another $40 billion. Total ecosystem, roughly $700 billion per year. Now, what happens to that ecosystem if fungi can eat plastic and turn it into soil? The waste management industry shrinks. If plastic biodegrades, you don't need to collect it, transport it, sort it, or bury it. The entire logistics chain simplifies. The recycling industry transforms. If biological enzymes can break plastic back into its original monomers, as the 2018 UK study demonstrated, then the expensive mechanical recycling infrastructure becomes obsolete. No more sorting facilities, no more specialized machinery, no more contamination problems. And the plastic production industry faces accountability. Right now, plastic producers externalize the cost of disposal. They make the plastic, they sell the plastic. Someone else deals with the waste. If a fungal solution exists that can be deployed at landfill scale and the plastic can be provably, cheaply, biologically destroyed, the political argument for banning single-use plastics becomes irresistible, and single-use plastics are a $400 billion per year market. Let me be clear about what I'm saying. I am not saying that Dow Chemical or ExxonMobil or any specific company sat in a room and said, suppress the mushroom research. That's not how this works. It never is. What happens is simpler and worse. The research exists, but the funding to scale it doesn't arrive. The grants go to incremental improvements in mechanical recycling, a system that currently recycles less than 9% of all plastic ever made. 9%. 91% of all plastic ever made is still sitting in a landfill, in the ocean, or in the environment. And the system that achieves 9% gets the funding, while the system that could achieve 100% gets published in a journal and forgotten. The EPA, the Environmental Protection Agency, has been aware of mycoremediation research since at least 2007, when Paul Stamets, the world's foremost mycologist, presented his work on using fungi to clean environmental contamination. Stamets demonstrated that oyster mushrooms could break down petroleum-based products, filter contaminated water, and restore dead soil. He presented this research directly to EPA officials. The response was polite interest, followed by no funding, no pilot program, no policy change, no deployment. Polite interest. That's how you bury a revolution without leaving fingerprints. If there's a hero in this story, it's Paul Stamets. Stamets has spent over 40 years studying fungi. He holds multiple patents on mycoremediation techniques. He's the author of 6 books. He's been a research partner with the Department of Defense, the NIH, and yes, the EPA. His work has demonstrated that fungi can break down nerve gas agents, that fungal extracts can treat smallpox and flu viruses, that mycelium networks can filter E. coli from contaminated water with 99% effectiveness, and that mushrooms can eat petrochemical waste, including plastics, and convert it to clean soil. In 2005, Stamets conducted a landmark experiment. He took a pile of diesel-contaminated soil so toxic that nothing would grow in it. He inoculated the soil with oyster mushroom mycelium. He covered it and walked away. 8 weeks later, the pile was covered in oyster mushrooms. Some were over 12 inches across. The mushrooms had broken down the hydrocarbons, the petroleum compounds, and metabolized them. When the mushrooms were tested, they contained no toxic residue. They were clean. They had converted poison into food. But here's the part of the experiment that changed everything. When the mushrooms fruited, they attracted insects. The insects laid eggs. Birds came to eat the insects. The birds dropped seeds. Within 16 weeks, the toxic waste pile had become a small, thriving ecosystem. Plants were growing. Insects were living in it. Birds were feeding on it. Life had returned to soil that had been dead and poisoned. The control pile, the one without mushrooms, was still toxic, still dead, still nothing. Stamets calls this mycoremediation, the use of fungi to remediate, to heal contaminated environments. He's been trying to get governments and industries to adopt it at scale for 2 decades. 20 years. The science is proven. The results are photographed. The mushrooms are available commercially. The process costs almost nothing. And the system still prefers to spend billions burying plastic in holes and pumping contaminated soil with chemicals. Because mushrooms can't be patented, mycelium can't be monopolized, and a solution that anyone can grow in their backyard doesn't generate consulting fees. Here's what you can do with this information today. Not theoretically, not someday, today. I'll start with the most accessible application and work up. Application 1. Mycelium composting. You can add mushroom spawn, specifically oyster mushroom spawn, to your regular compost pile. Oyster mushrooms are aggressive decomposers. They break down lignin, cellulose, and complex hydrocarbons that bacteria alone struggle with. A compost pile inoculated with oyster mushroom mycelium will decompose faster, more completely, and produce a richer finished product. Oyster mushroom spawn is available online for $5 to $15. You mix it into your pile. The mycelium does the rest. Application 2. Contaminated soil remediation. If you have soil that's been contaminated by petroleum products, an old oil spill, diesel contamination, parking lot runoff, automotive waste, you can treat it with oyster mushroom mycelium. Dig the contaminated soil into a pile or raised bed. Mix in fresh straw or wood chips as a food source for the mycelium. Inoculate with oyster mushroom spawn. Cover with a tarp to maintain moisture. Check periodically. Within 8 to 16 weeks, the mushrooms will have metabolized the hydrocarbons. The soil will be clean. You can test it. You can grow food in it. Application 3. Gray water filtration. Stamets has demonstrated that mycelium can filter contaminated water. A simple system, a burlap sack filled with wood chips inoculated with oyster mushroom mycelium, placed in a drainage channel, can remove E. coli, coliform bacteria, and chemical contaminants from water runoff. This is called a mycofiltration system. It's been tested at farm scale. It works. The materials cost almost nothing. Application 4. Plastic biodegradation. This is the frontier. You won't solve the plastic crisis in your backyard, but you can begin experimenting. Pestalotiopsis microspora, the Yale plastic-eating fungus, is not commercially available in spawn form, but oyster mushrooms and other common fungi have demonstrated degradation of certain plastics, particularly polyurethane-based products. Researchers have had success burying plastic products in actively colonized mycelium beds. The degradation is slow at the home scale, months to years depending on the plastic type, but it's real. And every bit of plastic eaten by fungi is plastic that doesn't sit in a landfill for 500 years. Application 5. Growing your own mycelium network. This is the big picture application. If you're a gardener, and if you're watching this channel, you probably are, you should be cultivating fungal networks in your soil intentionally. Mycorrhizal fungi form symbiotic relationships with plant roots. They extend the root system's reach by hundreds of times. They transfer nutrients between plants. They improve drought resistance. They suppress disease. You can purchase mycorrhizal inoculant, a powder containing fungal spores, and apply it directly to your soil or root zone when planting. One application establishes a network that persists for years. Your plants will grow stronger, your soil will become more alive, and you'll be building the same underground infrastructure that forests have used for 400 million years. 5 applications: oyster mushroom spawn, mycorrhizal inoculant, some straw, some wood chips. Total cost, under $30. Total potential, the remediation of the most persistent pollutant in human history. The mushrooms are ready. They've been ready. They were ready before we even knew we needed them. I want you to think about something. Right now, this minute, there are 8 billion tons of plastic on this planet that will never decompose on its own. 8 billion tons, sitting in landfills, floating in oceans, buried in soil, breaking into microplastics that are now found in human blood, breast milk, and the placentas of unborn children. Microplastics have been found at the summit of Mount Everest, at the bottom of the Mariana Trench, in Antarctic ice cores. There is no place left on Earth that plastic has not contaminated. And in the soil beneath our feet, in the forests, in the compost heaps, in the rotting logs and leaf litter of every ecosystem on the planet, are fungi that can eat it, that have been evolving the enzymes to eat it, that are waiting for us to stop ignoring them and start feeding them. The research has been published for 13 years. The mechanisms are understood. The organisms are identified, over 400 species. The practical applications are documented. The cost is nearly zero, and the world is still spending billions on mechanical recycling that captures 9%, still spending billions on landfills that store the problem forever, still spending billions on incineration that turns solid pollution into air pollution, still spending billions on cleanup efforts that can't keep pace with production. The system isn't solving the problem. The system is managing the problem. Because a solved problem doesn't generate revenue. A managed problem generates $700 billion per year, forever. But fungi don't care about revenue models. Fungi don't read industry reports. Fungi don't attend shareholder meetings. Fungi do what they've done for 400 million years. They break things down. They return nutrients to the soil. They close the loop that human industry keeps trying to break open. Every video on this channel tells the same story. A solution that works, that's free, that's proven, that's ancient or obvious or both, and that was buried because it threatened something more profitable. A pattern is the same every time. The solution exists, the system ignores it, and the only thing that breaks the cycle is you, knowing, doing, sharing, growing. The mushrooms are ready. They've always been ready. Feed it. If this changed what you think is possible, share it with someone who's been told the plastic crisis is unsolvable.